Scientists just reversed aging. Seriously. And no, I'm not talking about some sci-fi movie plot here. The research is actually published. The clinical data is absolutely staggering. And inside top-tier laboratories right now, biological clocks are literally spinning backward.
So, what if aging isn't this inevitable destiny we've all been sold, but a curable cellular disease? For centuries, we've just accepted physical decay as an unstoppable force. Well, today, we're going to completely shatter that assumption.
Let's jump right into part 1, the disease of aging.
To cure any disease, right, you first have to understand exactly how it works. In a massive landmark paper published in the journal Cell, researchers essentially mapped out the decay of the human body into very specific treatable mechanisms. They call these the hallmarks of aging. We're talking about things like genomic instability, mitochondrial dysfunction, and the dangerous buildup of toxic cellular waste. So aging is no longer this vague, unstoppable mystery. It's actually a measurable, finite checklist of cellular errors.
Okay, let's make this really concrete. Think of your biology exactly like a computer. Your DNA, that's the hardware. It's a pristine digital blueprint that largely stays perfectly intact your entire life. But your epigenome is the analog software. It's the operating system actively reading that DNA and telling your cells exactly what to do. So aging isn't hardware damage to your DNA. Aging is a corrupted hard drive. It's glitchy software corruption in your epigenome caused by decades of microscopic wear and tear.
Moving on to part 2, purging the zombie cells.
Now, what exactly happens when those corrupted cells refuse to die? They just linger around in your body, pumping out toxic inflammation. You essentially get a biological zombie apocalypse. But enter a brand new class of drugs called Senolytics. In plain English, these act as your body's targeted biological cleanup crew. They are compounds specifically designed to seek out and destroy these highly inflammatory, senescent zombie cells, and they entirely bypass your healthy tissue while doing it.
And when scientists put this to the test, the data is absolutely staggering. One major study took a specific senolytic cocktail, dasatinib and quercetin, and used it to clear out these zombie cells in aging mice. The results? It dramatically increased their physical function, completely restored endurance, and boosted their post-treatment survival by a massive 36%. We're not just managing symptoms anymore, guys. We're actively purging the decay.
Which brings us to part 3, the biological factory reset. Let's ask an even bigger question. What if we didn't just clear out the dead cells, but ran a literal factory reset on the living ones? This brings us to Dr. David Sinclair's breakthrough paper in Nature out of Harvard. His team figured out how to safely deliver 3 specific youth genes, known as OSK, into the cells of living, aging animals using a viral delivery system. They achieved something the scientific community historically thought was impossible, reprogramming cells to act young again without erasing their core identity.
The absolutely crucial part here is how this works on a molecular level. When those OSK genes are introduced, they activate specific enzymes called TET1 and TET2. And these enzymes trigger a process known as active DNA demethylation. Think of this as actively scrubbing away all that accumulated, age-related epigenetic noise on your software. This active DNA demethylation is absolutely essential for cellular regeneration and tissue repair. By erasing that chemical noise, the cell can perfectly read its original blueprint again, completely restoring its youthful gene expression patterns.
And this process leads to some miraculous, real-world physiological changes. Normally, when you observe a severed optic nerve in an aging mouse, there's zero recovery, none. But after administering this OSK factory reset, the optic nerve axons actually physically regenerated. The treatment fully restored visual acuity in older mice and those suffering from glaucoma. They didn't just slow down the blindness, they literally reversed it by restoring the youthful regenerative capacity of those neurons.
All right, part 4, actionable protocols for today.
So that is mind-blowing lab science. But how do we bridge the gap between Harvard labs and your daily life right now? Well, the foundation of longevity actually relies on something called hormesis, applying brief, intense periods of good stress to the body. Everyday practices like intermittent fasting, cold exposure, and high-intensity exercise basically trick your body into survival mode. This activates ancient nutrient-sensing pathways like AMPK and longevity proteins known as sirtuins. It literally forces your cells to conserve energy, clear out cellular waste, and aggressively repair themselves.
On top of that, we now have molecular tools that biochemically mimic the effects of fasting and stress to hit the pause button on cellular decay. Take NMN, for example. It acts as a booster for NAD+, which is a critical molecule that fuels those sirtuin repair enzymes we just talked about, giving them the energy they need to fix DNA damage. On the flip side, drugs like rapamycin inhibit the MTR pathway. That's your body's central growth and nutrient sensor. It essentially tells your cells to stop dividing, hunker down, and start cleaning house through a process called autophagy.
And listen, this isn't just theoretical science confined to a petri dish anymore. The Dog Aging Project, backed by a $7 million grant from the National Institute on Aging, is currently running the Triad clinical trial. They're testing rapamycin right now on normally aging companion dogs, animals that share our homes, our stress, and our environments, to measure lifespan and health span extension outside the lab. Real-world translation is happening as we speak.
Let's look at part 5, the future of longevity.
So, where is all of this actually going? Let's look forward to the rapidly approaching horizon of human longevity tech. Based on the current trajectory of these massive studies, here is a highly realistic timeline. By 2026, we're going to see conclusive triad dog data and early aggressive human trials for longevity therapeutics. By 2028, we can expect to see refined FDA-approved Senolytics available for targeting specific age-related diseases. And by 2030, the first mainstream localized cellular reprogramming applications exactly like what Sinclair did for vision could hit human clinics. That is going to fundamentally change modern medicine forever.
The science of aging is literally rewriting the fundamental rules of human biology. But the ultimate question is, can a human being actually reverse their biological age today? Well, tech entrepreneur Brian Johnson just released data showing a cumulative biological age reduction of 5.1 years across all his major organs. In our next explainer, we're going to decode his exact don't die blueprint protocol step by step to see exactly how he achieved those numbers. So make sure to subscribe right now to catch it, and I'll see you in the next one.